Nucleic acids are our last type of macromolecule that we want to discuss. The last type of macromolecule we find in you. And we see this really three major forms in you. We see DNA, RNA, and ATP, or our cell energy. So when we build these guys, we're going to build them all from nucleotides. Nucleotides have a very basic structure that have a phosphate, a base. The base is A, C, T, or G. Or if you're talking about RNA, it's A, C, U, or G. And this is a sugar. It's pretty simple, right? There's not much to it. We're going to take this monomer unit and put a bunch of them together to make DNA, RNA, and ATP. So this sugar with the phosphate, and in this case, the adenine or A base associated with it, and we're going to build these. We build them into DNA, we build them into RNA, and we build them into that ATP. All of these are information and energy storage types of molecules. DNA and RNA in particular are information storage molecules. So the DNA we have talked about before, and we will talk about much more in the future, makes up your chromosomes inside the nucleus, inside your cell. We can look at them as making up genes, right? RNA, these are actually going to be critical for you for protein production. I'll show you why later on. And we also look at them as having a very significant role in other processes in your body that we'll get to. So this polynucleotide, again, it's a dehydration reaction. Are you getting this? If you're building something, you're doing a dehydration reaction. So is water used or formed? Well, it's dehydration. You're making water, right? You're forming water. So to build these together in a DNA structure, here's our basic simplified structure of DNA. You have a sugar and phosphate backbone. We showed you that. So it's sugar, phosphate, sugar, phosphate, sugar, phosphate. And attached to those are the bases that we talked about, the nitrogenous bases. Nitrogenous because they're based in nitrogen. Okay. And when we look at building DNA, we're actually going to build something known as a double helix, which many of you may already know. We're going to take two polynucleotide strands and stick them together so it looks like this. Now, when we do this, and we'll talk much more about this later on, what we have to think about is that to build these and to have the sugar and phosphate and nucleotides coming together, bases coming together into these nucleotides, we think about DNA as being double-stranded. The hydrogen bonds form in between. If we go back here 1 second, those dotted lines, remember, indicate hydrogen bonds. We saw this in the chemistry lectures. If you don't remember what a hydrogen bond is, go back and look at that. So we get those hydrogen bonds. A's and T's pair up, C's and G's pair up when we talk about DNA. Again, we're going to spend an entire lecture on thinking about DNA and then another one thinking about how it replicates and behaves. So we'll come back to this. RNA is single-stranded most of the time. But what we have to think about is the interaction between these two. DNA is your information unit stored in your nucleus, and it never leaves the nucleus. RNA is more like the messenger or the worker. We're going to look at it as picking up information from your DNA. The RNA carries that message, and then it forms a protein. So this is what is referred to as the central dogma of genetics. DNA gives information to RNA. RNA directs all of the mechanics in your cells to make proteins. So when we look at these guys, we're going to look at the genes, or the part of your chromosomes in your nucleus directing the building of all of these macromolecules, particularly looking at the directing of building polynucleotides, like we said. It has to build itself. We're going to build polypeptides. We're going to get instruction on how to build carbohydrates, how to build lipids. All of this information is coming from the instruction book that is your DNA. Okay? So what we're going to look at when we get into genes and thinking about the production of proteins, we're actually going to take information from DNA, like we said, give it to RNA through a process known as transcription. We're going to take RNA and build proteins through something known as translation. So these will be integral to our discussions on things like, when you can't break down lactose, why not? Okay? These will be very important discussions. If you can't build lactase, the enzyme, if this is lactase, it's not, it's too small. But if this was lactase, if you can't build this, what might have gone wrong? Well, maybe the RNA is bad. Maybe the DNA is bad. Maybe the process is bad. But most of the time what we see is there's some mutation in the gene that stops the protein from being formed. You don't form lactase. You can't break down lactose. Now you're lactose intolerant.